All right, so this week we have translation. And so we'll start with talking a little bit about transcription. So in my transcription example, I have the minecart going around, and every time the minecart goes over a detector rail, it triggers a code block, and that code block reads the base next to it, which is DNA, and then it makes an mRNA base, and then I have a piston that automatically pushes it up. So I'm making a growing chain of mRNA molecules, and then up here I have basically a bend so that I could get another angle. And so it's pushing the mRNA blocks into my ribosome. And so in my ribosome, I'm reading the set of 3 blocks that are adjacent to these 4 blocks. So those are the 4 types of codons that I could have in my setup. And then I have code blocks to read those, and if what they see is identical to this, then they make the appropriate amino acid over here. And so as my amino acids are made, they can only be like 20 blocks long, then they get pushed out. And so I have a growing chain of amino acids. So the wooden blocks of different types are my amino acids, my growing chain of amino acids. And as those amino acids are assembled, then they're making a polypeptide. So that's my example of my ribosome. So literally what happens in the ribosome is that I have a codon that enters the A site, and then that is read by a tRNA, and that tRNA carries the appropriate amino acid, and then those amino acids get assembled into a protein. So to review some of our terms, so last week we had transcription, and so we talked about initiation, elongation, and termination of transcription. We have those same processes in translation. We have initiation, elongation, and termination, and they refer to the same sorts of processes. In initiation, my mRNA is binding to my ribosome. In elongation, I'm making a growing chain of amino acids, and in termination, I'm stopping the whole process. But we have a little bit more complexity in that in translation, we have 3 types of RNA that are involved, and all 3 of these types of RNA are transcribed by different parts of the genome. And so we make mRNA, and from that mRNA, we're going to make polypeptides. We make tRNA, and our tRNA is our go-between between mRNA and amino acids, and we make rRNA, and rRNA is our ribosome. So it's our ribozyme. It's going to enzymatically allow us to connect those amino acids together. So last week we talked a little bit about mRNA. We have our 5' cap and our poly A tail, and those are our untranslated regions of our mRNA. So they get transcribed, but they don't get translated. Instead, they serve to allow our mRNA to be successfully translated. So our 5' cap protects the 5' end, and it facilitates binding to the small subunit of the ribosome. My poly A tail protects my 3' end from getting consumed by exonucleases. And so I have some regions also in my untranslated regions, and one of those is the Shine-Dalgarno sequence if I'm talking about a prokaryote, or the Kozak sequence if I'm talking about a eukaryote. And the Shine-Dalgarno sequence is at the front. It's in the 5' untranslated region, and it recognizes the small subunit. So if I have my ribosome, those are my 3 sites of my large subunit, and then this is my small subunit. So there are, right, this is RNA and proteins, and in my small subunits, so I have a series of bases, RNA bases, and my Shine-Dalgarno sequence recognizes the bases in the RNA. So that comprises initiation in prokaryotes. The process of my Shine-Dalgarno recognizing my small subunit and binding to it. All right, that is initiation. And in eukaryotes, it's more complicated, but in essence, we have other sequences and other mechanisms for our mRNA to recognize that small subunit. And in eukaryotes, the Kozak sequence tells our ribosome where to start transcribe, or where to start translating, where to start making amino acids. And so within our mRNA, within the coding region, so within the region that gets translated, we have sense codons and nonsense codons. So sense codons bring back an amino acid, so they're involved in elongation, and nonsense codons don't bring back an amino acid, so they're involved with termination. So as far as our tRNAs go, the truly translational protein RNA structure is amino-acyl tRNA synthetase, and it recognizes specific amino acids, and then it pairs them with specific codons. So that allows our tRNAs, which are charged with amino acids, if they have an amino acid, to bring those amino acids into the ribosome. And then they lose those amino acids in the ribosome and they become uncharged. And every time they're charged, they have the appropriate amino acid added to their acceptor stem. So there are regions within the cloverleaf shape of each individual tRNA molecule that allows tRNA synthetase to recognize the appropriate amino acid. But all of their acceptor stems are the same. So once it recognizes the appropriate amino acid, it can charge it by adding it to the acceptor stem. And so our rRNA is ribosomal RNA, and that makes up our ribosome. So our rRNA, or RNA in general, often has enzymatic properties, and when it does, we say that it functions as a ribozyme. So RNA can store genetic information, but it can also function as a protein, structural protein, enzymatic protein. It can do all kinds of things. And so our ribosome acts as a ribozyme. There's parts of it that recognize other mRNA molecules, other DNA molecules, and it binds to them, and there are parts of it that function enzymatically. So we have our small subunit, which is strictly involved in initiation, recognizing that mRNA molecule, and our large subunit, and that's where all of the enzymatic activity happens. And so we have our A site, which is our amino-acyl site, our P site, which is our peptidyl site, and our E site, which is our exit site. And so as our RNA moves through the ribosome, it translocates. So each step of the way, 3 bases translocate and move to the next step. And that process is called elongation. So we're going to describe elongation before we get to termination. So if I have an mRNA molecule, it's going to start with AUG, and that is going to go into the A site, and that is going to bring back methionine. And then that AUG is going to translocate to the P site, and it's going to take that methionine with it. And then the next base, for simplicity, we'll just say the next base is also methionine, comes into the A site, and then the tRNA brings in methionine to the A site, and now we have the opportunity for our P site to do its thing. So the P site is going to create a peptide bond between those 2 amino acids. And so we have our first peptide bond, and the P site is the peptidyl site because it catalyzes that reaction. And then the next thing that happens, actually, I'm going to add my tRNA molecule here. So UAC. So then I'm going to have another translocation event. And so what's going to happen is that this methionine is going to come up out of the ribosome. This methionine goes into my P site, we'll just say, for simplicity's sake, we'll say that we have another methionine coming in. And my tRNAs are going to move also. And then I'm going to cleave this peptide bond. And so it's important to note that my peptide chain, my amino acids, are growing out of the P site. And so when that tRNA, that UAC tRNA, gets to the E site, it doesn't have its amino acid anymore. It gets stripped off when that peptide bond forms. And so we call it the E site because that's where uncharged tRNAs leave the ribosome. So my uncharged tRNA leaves the ribosome at my E site, and then my mRNA is just going to keep feeding through the ribosome. Right, so that describes translocation and the process of going from the A to the P to the E, which gives us elongation, our growing chain of amino acids, or our growing polypeptide. And that process continues until we get to a nonsense codon. So our nonsense codons don't have an amino acid associated with them. So we'll imagine that our next amino acid, our next codon is UAG, which is a stop codon or a nonsense codon. So I'm going to have all these guys move one position, and he's going to move into the A site. And then same deal, these guys are going to move up and out. All right, so now we've reached the point where there is an absence, so I'm just going to put an X there, because there is no tRNA that carries an amino acid that recognizes UAG, so nothing comes into the A site. So my P site is trying really hard to make this peptide bond, but it can't because there's no amino acid in the A site. And so when it tries and fails, we have another molecule called our release factor. That comes in and it dissociates the entire ribosome. So it pulls away the small subunit and it pulls away the large subunit. And that causes my polypeptide to leave the ribosome. So my polypeptide only has 3 bases. So I'm just going to redraw those here. So that is my nascent polypeptide. It happens because of the release factor that enters when I don't have amino acid to add to my growing chain. And so that is my polypeptide, right? That's the goal of this entire process. And so just like DNA and RNA have polarity, they have 5' ends and 3' ends, so does my polypeptide. So the end at the front is called my amino end, and it's end primed, and then my other end associated with the 3' end of my RNA is my carboxyl end. So my amino and my carboxyl ends of my amino acid, so the front and the back. And then the last thing that's going to happen is post-translational modification. So seldom is that nascent polypeptide in in the form that I want it to be in its functional form. So I'm going to add molecules along the the body of the polypeptide. I'm going to change the polarity along the polypeptide. I'm going to change how it interacts with itself. And that's going to dictate the secondary structure of that polypeptide. So I'm going to have some functional motifs like alpha helices and beta barrels. It's going to determine the final tertiary structure, and so those are functional domains, so that those domains are going to allow it to interact with other molecules and other proteins, and those domains are where the enzymatic activity is going to happen, and it's going to dictate how the three-dimensional structure of that protein that I just made is going to interact with other three-dimensional structures, forming quaternary structure. So the post-translational modification, some of which happens via molecules we call chaperones, is very important to the final functional role of our protein. And that's it for transcription and translation.